Good evening, everyone. Today we are going to start with discussion of week 4 lecture content of immunology. So, I'm sharing my screen. So, today is the week 4 of host pathogen interaction. My name is Chatterjee. I'm a graduate student in IISc Bangalore. So, I am one of two TAs for this course. So in this class, it was discussed that what is innate immunity. Okay, so there is a survival of the fittest theory, and where professor has described about this, and that for the if you want to be fit, your immune system is also needed to be good, right? So because of there are different pathogens are surrounded us. So so the survival of the fittest theory, as we all know, that was described by British naturalist Charles Darwin. So there are different kinds of bacteria, viruses, fungi surrounding us. So this is a representative scanning electron microscope image of a large number of bacteria. These greens are bacteria in the gingiva. Gingiva means gums. In the gums of a human mouth. Could you imagine the amount of bacteria residing in our body? Okay, in a human mouth, this number of bacteria are there. So there should be some defense system of us which will protect us against this, right? So we have this innate immune system. So as you can see, so is the human body is a human body human? No. Why? So basically it's trying to say that it's the whole human body consists of like humanic cells, like just a basic way of saying. Why? Because microbes are 10 times more the number of human cells. Understood? So from the head to toe we are human, so the cells are also human cells. But no, it's not like that, because microbes are there at 10 times more than number of human cells. So 9.1% human cells, bacteria are 1.3 times more than number of human cells. Okay, and 43.5% are human cells. This estimation does not include fungi, viruses and phages, and it will be more, at least an order of magnitude, and it will come around 9.5%. So you can imagine that in human body there are a lot of other microbes residing. Additionally, genes are approximately 200 times more than the host genes. Genes are approximately 200 times more than the host genes. Human microbiome, around this many genes are there, and human genomes are around 23,000 genes. So how host microbes are associated? The microbes and host associated are not always toxic. Okay, like it can be, it is, there are things where you can see that mutualism is there, where we need those good bacteria in our body, but there are also different viruses, bacteria, pathogens which you don't want. So this kind of interactions, you can divide into three things. One is commensalism, one is mutualism, and another one is parasitism. So commensalism means that the bacteria or virus whichever is staying with you is not harming you, but it's not harming, you are happy as it is, but he is a kind of on evil side, that he is just making use of your cells and your nutrients and all, and he is getting benefited, but you are not getting benefited for him, like for that microbe. But the thing that the bacteria or microbe I'm saying here is like it is doing that. So for the mutualism, what is happening, that the bacteria or any microbes which is staying in host, it is like the host as well as the microbe is getting benefits or getting like help from each other, right? So that is mutualism. And in parasitism, the host body or the human cells are getting uh, infected, affected by those microbes. So microbes are happy, but because of that, because they're using our host cells, but the host cells is getting infected or affected. Innate immunity evolving with pathogen. Sorry. So there are MSC, immunoglobulin, TCR, RAC, lymphocytes. So there are different receptors, toll-like receptors, complement system, phagocytosis, antimicrobial peptides. So innate immunity is there, and then you have your different B cells, T cells. So B cells will provide antibodies. So these antibodies and all are coming under adaptive immunity. So you are getting used to the microbes, and then your body is generating antibodies against it. So innate immune genes evolve slowly while pathogens evolve rapidly. So many of host restriction factors undergo positive selection to resist the evading mechanism of pathogens. So many of the host restriction factor, what it does, this is host, this is pathogen. So undergo positive selection to resist the evading mechanisms of pathogens. So innate immunity evolved 700 million years ago. Since the origin of sponges, adaptive immune system evolved 450 million years since the first jawed vertebrates. So there are also faster evolution of microbes, which results to innate immunity as a first line of defense against plethora of microbes. So how does pathogen get entered into our body, and then what is the immune responses? So pathogen gets entered into our body, containment by a anatomic barrier. So we do have an anatomic barrier that can be skin, that can be your mucosal layer inside mouth. So a containment by an anatomic barrier which prevents infection. But if this barrier or this first line of defense gets failed, then we get infected. After infection, what happens? Recognition by performed non-specific and broadly specific effectors. Recognition by preformed non-specific and broadly specific effectors, which is removal of infectious agent. Then if this recognition by preformed non-specific and broadly specific effectors gets failed, what happened? Recruitment of effector cells. Okay, so this is early induced innate response, which is seen in between 4 to 96 hours. Then recognition of PAMs, activation of effector cells and inflammation, removal of infectious agent. So if the R cells can recognize PAMs and activation of effector cells and inflammation happen, in that stage also removal of infectious agent can be possible. But again, if R cell or we fail at this step, what happens? The transport of antigen to lymphoid organ. Then here, after transporting, the adaptive immune response starts playing action. So then recognition of knife, B and T cells happen, clonal expansion and differentiation happens, and then ultimately those antibodies will affect against those microbes, and then removal of infectious agent has been done. So then what are the components of innate immunity? Components of innate immunity: physical, mechanical, anatomical barriers, biochemical, chemical barriers, antimicrobial peptides, proteins. Examples are, so there are antimicrobial peptides and proteins. So there are different sugar binding proteins, C-type lectin receptors, and then mannose binding protein, maltose binding proteins. So MPP, CRP, APP, serum amyloid, antitrypsin. So these are examples which are antimicrobial peptides or proteins. Complements: microRNAs against viruses, microbial, microbiological barriers, cellular, almost all cells, immune and even non-immune cells, particularly NK cells, macrophages, dendritic cells expressing PRR, sensing products of metabolic pathways unique to a particular class of microbes. Mechanical, chemical and microbial barriers. So mechanical, what are the mechanical barriers? So there are skin, gut, lungs, eyes, and nose or oral cavities. So mechanical barriers, epithelial cells joined by tight junctions, longitudinal flow of air, uh, air or fluid, longitudinal flow of air or fluid, movement of mucus by cilia, tears, nasal clia. Chemical barrier in skin, fatty acid, beta defenses, lamellar bodies, gut chemical chemical barriers are low pH, and then enzymes, lungs, pulmonary surfactant, eyes, nose, enzymes in tears and saliva, beta defenses, lamellar bodies, cathelicidin, gut, alpha defenses, cryptidins, reg 3, lungs, alpha defenses, and then eyes or nose and oral cavity, histatins and beta defenses. Microbiological barrier are normal microbiota. Okay. Skin physical components of defense. So epidermis, which is the outer layer composed of multiple layers of tightly packed cells. Few pathogens can penetrate these layers. Shading of dead skin cells removes attached microbes. Epidermal dendritic cells, phagocytis pathogens. These cells extend out among other cells of the epidermis, forming a network to intercept invaders. So, then skin physical components of defense. So skin is, as we know, the skin is a physical components of defense. So the epidermis, the outer layer, composed of multiple layers of tightly packed cells. Okay, so few pathogens can penetrate these layers, but shading of dead skin cells remove attached microbiome. So our skins have layer of dead skin cells, right? So when those get sheds off, whatever microbes are attached, they will also get shed off. Epidermal dendritic cells, phagocytis pathogens. These cells extend out among other cells of the epidermis, forming a network to intercept invaders. Dermis contains protein fibers called collagen. So gives skin strength and pliability to resist abrasions that could introduce microorganisms. So dermis contains protein fibers called collagen, give skin strength and pliability to resist abrasions that could introduce microorganism. Skin, which is a biochemical components of defensin, perspiration secreted by sweat glands. Salt inhibits growth of pathogen by drawing water from their cells. Antimicrobial peptides, defensin and cathelicidins, lysozyme destroys cell wall of bacteria. Okay, so perspiration secreted by sweat glands. There you can see that salt, which inhibits the growth of pathogen by drawing water from their cells. Antimicrobial peptides are present there, example defensins and cathelicidins. Lysozymes present there, which destroy cell wall of bacteria. Sebum secreted by sebaceous oil or oil glands helps in keeping skin pliable and less likely to break or tear. Lowers pH of skin to a level inhibitory to many bacteria. Skin physical or biochemical components. Epidermis of skin. So this is the stratum corneum layer, and then stratum lucidum layer, stratum granulosum, stratum spinosum, stratum basal. So this is the enlarged view of the stratum granulosum and stratum spinosum, where you can see watertight lipid layer, lamellar bodies, beta defensins and cathelicidins are present there. So mucosal surfaces line all body cavities open. So mucosal surfaces are present. They line all body cavities which are open to the outside environment. Unlike surface epidermal cells, epithelial cells are living and closely associated with the lymphoid tissues, where antigens are transferred to M cells. Epithelial cells pack tightly to prevent entry of pathogens, but often only one cell layer thick, so pathogens sometimes breach the barrier. Continual shedding of cells carries attached microorganism away. Besides producing mucus, mucous membranes also produce lysozyme and other antimicrobial peptides. Every day, an adult human swallow and digest about 1 liter of mucus. So mucosal surface, just lungs, secrete mucus, helps in trapping and ejecting microbes. This bronchial ciliated epithelium. These are goblet cells, these are cilia, these are blood vessels, gland duct, mucous glands. So this mucous glands, and from there it gets secreted, mucus gets secreted and helps in trapping and ejecting microbes. So cilia are there like to flow the fluid or mucosal fluid through the cells. So this is one another mucosal surface which is present in gut epithelium. This is the example which is present in bronchial ciliated epithelium. So in the gut epithelium you won't find cilia. Okay, so here we will see that villi like appearance. So in the gut you know that the villi are present, right? So we'll see goblet cells, and then when microbes come out, like microbes will come, and then what happens that you have paneth cells. You have paneth cells here. It will produce alpha defensins, reg 3 like components, and macrophage are also there. So then these are the like mucosal element which will fight against microbes. Okay, any question? So lysozyme is another antibacterial component which acts so this is which acts as in bacteria. So it's a gram positive bacteria, it's a gram negative bacteria. So there are different protein there are different layers of peptidoglycans. So it basically cleaves bond in the peptidoglycans between sugars. Okay, this is how it acts against bacteria. So we will discuss defensins, catholicidins and histatins are activated by proteolysis to release an amphipathic antimicrobial peptide. If you have any doubt, we'll just discuss that and then we'll start with this chemical components. Thanks, Zubin. So, defensins, catholicidins, and histatins are activated by proteolysis to release an amphipathic antimicrobial peptide. So this is alpha defensins, beta defensins, cathelicidins, and reg3 lactocidins. So alpha defensins, there is a pro region and there is a cut region in between AMPH and pro region. So basically it's a domain which is the active part of this components. So there you will these are getting activated by proteolysis to release an amphipathic antimicrobial peptide. Okay, amphipathic antimicrobial peptide. So defensins, there are three subfamilies of defensins: alpha, beta, and theta defensins are distinguished on the basis of amino acid sequences. So each family has members with distinct activities, some being active against gram-positive bacteria and some against gram-negative bacteria, while others are specific for fungal pathogens. Okay, so electrostatic attraction and the transmembrane electric field bring the defensin into the lipid bilayer. So if there is a like electrostatic attraction and the transmembrane electric field, so if there is a transmembrane electric field, so that will bring defensin into the lipid bilayer. So defensin peptides form a pore, and you will see that there are three subfamilies of defensins: alpha, beta, and theta defensins are distinguished on the basis of amino acid sequence. Each family has members with distinct activities, some being active against gram-positive bacteria, some against gram-negative bacteria. So some more antimicrobial protein or peptides, example lactoferrin, sequester Fe+2. So lactoferrin is a potent antiviral, antifungal, and antiparasitic activity towards a broad spectrum of species. In bacteria, it has bacteriostatic properties. To counter its action, bacteria has siderophores. In gram-negative bacteria, it has bactericidal activity due to its binding to the lipid A part of the bacterial LPS. Okay. What does azurosidine do? An antimicrobial and chemotactic protein and inactive serine protease, bacterial permeability inducing protein, vitamin B12 binding protein. So these are different other like some more antimicrobial proteins which help in innate immunity. Biochemical barriers, reactive nitrogen species, which is NO with the nitric oxide, nitrogen dioxide NO2, and peroxynitride. Synthesis from arginine by NO synthase. So there are different reactive species, RNS, ROS, and so it acts as like in the stress condition. So reactive oxygen species will be saying that if something is like not going good, like you are in stress, your cells are in stress, so they start releasing those as a signal, and they are also acting as a biochemical barriers. Microbiological barriers. So ubiquitous commensal bacteria, lactobacillus and clostridium species, Bacteroides fragilis, Bifidobacterium species, Escherichia coli, Firmicutes, Bacteroidetes, Actinobacteria, Proteobacteria, other phyla. So in mouth you can see that, so these are the color coded graph where you can see in mouth there are ubiquitous commensal bacteria are present, that is lactobacillus, clostridium, and different other some fractions of different other bacteria. In skin you can see that mainly actinobacteria are present. In in gut you will, in stomach and gut you will find ubiquitous as well as other little bit, but in esophagus you will again find ubiquitous commensal bacteria. So there are different studies have been done to find innate immune system in different species. So toll-like receptors has been studied in Drosophila. So you can see there is a receptor toll, and then from fungi you can see that this is getting activated, and after downstream signaling you will see that antifungal peptides gene is getting activated. Okay. So there are different papers which suggest that the dorsoventral regulatory gene cassette, Spagel or toll or cactus controls a potent antifungal response in Drosophila adults. A human homologue of the Drosophila toll protein signals activation of adaptive immunity, defective LPS signaling in some mice. You can see that if like in some mice you have seen, if you mutate TLR4 gene, which is a toll like receptor gene, you will see defective LPS signaling. So it depicts that all these are important for innate immune system. So innate immune response in Drosophila: cells for defense, fat body cells and hemocytes are phagocytic cells that act as a part of immune system. Antimicrobial peptide, drosomycin, DIF dependent, diptydicin, atacin and secropin, release dependent via immunodeficiency signaling pathway. Microbial sensors in Drosophila: peptidoglycan recognition proteins, PGRPs, 13 genes and bind to the peptidoglycan of bacterial cell wall. Gram-negative binding protein recognizes LPS and beta-1,3 linked glucans of gram-negative bacteria, recognizes fungi. GNBP1 and PGRPSA involved in recognition of peptidoglycan from gram-positive bacteria. Downstream signaling through sensors in Drosophila: so it activate serine protease called GRAS and trigger proteolytic cascade that terminates in the cleavage of protein splagel. So there is homodimerization of spagel and leads to, so the TIR domains of toll recruit the adapter MYD88, which activates. So TIR domain activates MYD88, which recruits MYD88, which activates the signaling pathway similar to NF-kappa beta pathway. Okay. So toll is a central to defense against both bacterial and fungal pathogens in Drosophila. Toll itself is not a pattern recognition receptor. The toll protein controls dorsal ventral patterning in Drosophila embryos and activates the transcription factor dorsal upon binding to its ligand spagel. So pattern recognition receptors or TLRs. Pattern recognition receptors, which is toll-like receptors, RIG1-like receptors, NOD-like receptors. So toll-like receptors recognize bacteria, fungi, viruses on surfaces and in endosomes of cells to induce pro-inflammatory cytokines and type 1 interferons. RIG1-like receptors, intracellular sensors, recognize RNA of RNA viruses and induce pro-inflammatory cytokines and type 1 IFNs. Not like receptors, intracellular sensors recognize wide range of PAMs to induce pro-inflammatory cytokines and involve in processing IL-1 family cytokines. So there are different pattern recognition receptors. So there are different toll-like receptors, TLR1, 2, 3, 4, 7, 9, like that, and they act at different downstream signaling. So they basically pass on the information that they have interacted with pathogens. Okay, the one example we talked in the activation of MID88 and then the activation of NF-kappa beta, and then it will induce pro-inflammatory cytokine, and then different cytokines will be produced and will help in forming inflammasome complex. So human toll or interleukin-1 receptor or TIR family, toll-like receptor subfamily, single pass transmembrane proteins, human toll-like receptors. 18 to 25 copies of a leucine-rich repeat, each composed of 20 to 25 amino acid. PAMs recognized by toll-like receptors. Triacyl lipopeptide TLR1 or 2, diacyl lipopeptide TLR2 by 6, LPS TLR4, flagellin TLR5, uropathogenic bacteria TLR11. So PAMs recognized by toll-like receptors. So these are the toll-like receptors, so which recognize like lipid lipoproteins. These are the receptors which recognize proteins, and these are the receptors which recognize DNA or RNA. So more specifically, triacyl lipopeptide by TLR1 or 2, diacyl lipopeptide by TLR2 or 2 by 6, like 2 or 6, lipopolysaccharide by TLR4, flagellin by TLR5, uropathogenic bacteria by TLR11, double stranded RNA TLR3, imidazole single stranded RNA TLR78, CpG DNA TLR9. Sorry. So innate immune recognition by mammalian toll-like receptors. So there are examples of different toll-like receptors, and their ligand and hematopoietic cellular distribution. So TLR1, TLR2 are heterodimer. So ligands are lipomannan. So you can just read this list. So ligands are lipomannans and like that. Some are double stranded RNA, some are LPS, and hematopoietic cellular distributions where they are present. Example TLR1 to 6, their presence on monocytes, dendritic cells, mast cells, eosinophils, and so on. TLR13, which is only mouse only present. So ligand are single stranded RNA, and hematopoietic cellular distributions are macrophages, dendritic cells. So again, this is the list of pattern recognition receptors. TLR signaling, MYD88 dependent signaling, Myelin differentiation primary response gene 88. This is a full form, which is present in macrophages, dendritic cell, plasmacytoid dendritic cells. TRIF dependent signaling, TIR containing adaptor inducing IFN gamma axon, like present on macrophages and dendritic cells. So MYD88 dependent signaling pathways, again like all those TLRs are getting activated or are recognizing different lipids, proteins, and then it's recruiting adaptor protein MYD88, which acts downstream by forming like TRAF6 IKK complex, and then acts on NF kappa beta. So there is inhibitory kappa beta which gets separated from NF kappa beta, and then NF kappa beta acts on the enters into the nucleus and acts on inflammatory axon producing inflammatory cytokine. TRIF dependent signaling, LPS TLR4 is LPS is recognized by TLR4. This is the same story. So where you can see that again NF kappa beta pathway, and then you can see MAPK are also getting involved where it's recruiting TIRF. So yes, so ultimately inflammatory cytokines are getting synthesized, and then type 1 interferons and interferon inducible genes are getting getting upregulated. So this is the summary of TLR and its adapter. So TLR2 or 1 adapter is MYD88, TLR3 adapter is TRIF, TLR4 MYD88, TLR5 again MYD. So mostly MYD88 is the adapter. So Yes, so sorry, inflammatory mediators and type 1 and type 3 interferon. So these are the examples of different inflammatory mediators. So vasoactive amines, example is histamines and serotonin. Um, then eicosanoids, thromboxanes, leukotrienes, prostaglandin, peptide bradykinin, proteins, IL-1 beta, interleukin 6, TNF alpha, type 1 interferons, IFN gamma consist of 13 members and single IFN beta. Type 1 interferon inducible genes are IP10, RANTES, etc. Type 3 interferons, interferon lambda consist of 3 members. Role of type 1 interferons in antiviral. So what happens? Oligoadenylate synthase, ADP2 2' to 5' linked oligomers, and then endoribonuclease RNase, and then viral RNA. So P1 kinases, it's acts on initiation factor and then translation of proteins. MX protein increase resistance to virus. Activate NK cells, kill viral infected cells. Adaptive immunity increases antibody and CD8 T cell responses. Increases MHC1 expression. Polymorphisms of TLR signaling molecules and their association with human disease. So in terms of function, we can see there are polymorphism. So what happens? So in this TLR4, if there is a mutation in like D299 to G, what will happen? Decreased response to inhaled LPS, decreased atherosclerosis, increased gram-negative bacterial infections, increased severity of atopy, decreased airway hyperreactivity. IRAC4 stop codons 287 293 increased gram positive infections. CD14, sorry. So what will happen, like it is seen that due to polymorphism, increased response to LPS and several other polymorphic activities there. So the association between TLR polymorphisms in human and disease susceptibility or protection. TLR or adapter protein, TLR polymorphism and human disease. So TLR2, TLR polymorphism, what will happen? T596 to C, R753 to Q, which is associated with a protective effect against leprosy and predisposition to tuberculosis, predisposition to staphylococcus infections. TLR4 several polymorphisms including D29 299G and T399I, which increases risk of systemic inflammatory syndrome and septic shock, decreased risk of atherosclerosis, no association with susceptibility or severity of rheumatoid arthritis. TLR5, a dominant negative stop codon mutation, increased susceptibility to pneumonia caused by Legionella pneumonia and negatively correlated with Crohn's disease. The association between TLR polymorphisms in humans and disease susceptibility to or protection. So, role of innate immunity during cancer. Way back in 1891, William B. Coley injected streptococcal in patients with inoperable cancer. Streptococcal infection helped in shrinkage of malignant tumor. Over the next 40 years, as head of the bone tumor service at Memorial Hospital in New York, Coley injected more than 1,000 cancer patients with bacteria and bacterial products. So William B. Coley is called as father of immunotherapy, inventors of Coley's toxin. So these bacteria or bacterial products were known as Coley's toxin and are used in treatment of bone and soft tissue sarcomas. Okay. So this is a list of the developmental status of compounds that target TLRs for viral and bacterial infections. So we will discuss week 3's assignments with you. The question is, what is the approximate number of hematopoietic stem cells in bone marrow? So it's around 1 in 50,000. What are the marker of hematopoietic stem cells? CD34 and SCA1. Okay. If the bone marrow cells are cultured with a factor called MCSF, it will differentiate into macrophage. To make PDCs from hematopoietic stem cells, which factor should be used? Factor FLT3. Which of the following immune cells is formed through both lymphoid and myeloid lineage? It's dendritic cells. If NPT test was performed and did not result in blue color complex formation, it is indicative of chronic granulomatous disease. Eosinophils are primary phagocytes having acidic granules, having multilobe nucleus, having major basic proteins, decreased during allergic reactions. So if you go to NPTEL lecture 3, yes. Yes, so eosinophil are stained by eosin, which is acidic dye and which is negatively charged. So what the cytoplasm of eosinophil contain? It is crystalloid in nature and it contains major basic protein. Okay, the name is major basic protein or MBP. So major basic protein consists of toxin for helminth, induced histamine from mast cells. So the answer is lecture 4. Eosinophil are having major basic protein. Which of the following statements is or are correct? Sorry, incorrect. The antibodies are predominantly produced by erythrocytes. Incorrect. On maturity, the red blood cells do not lack a nucleus, which is also incorrect because we know on maturity RB cells lack nucleus. The white blood cells have a lesser lifespan compared to the red blood cells. And this is true. Which of the following statements are correct? Macrophages mature into monocytes and migrate from blood to tissues. This is the other way around. Monocytes mature to macrophage and migrate to blood to tissues. B lymphocytes come under the granulocyte category. False. Megakaryocytes are produced from the blood platelets and released into the bloodstream. This is also false. Which of the following statements are correct? Neutrophils and basophils are predominantly involved in the phagocytosis of pathogens. No. Leukocytosis associated with a decrease in the neutrophils upon infection. Leukocytosis is no, this is also not correct. Chronic granulomatous disease is a genetic disorder. This is correct. So if you see leukocytosis, yeah, leukocytosis or diabetes is increase in neutrophils in circulation up infection. After infection, the increment is called leukocytosis. Okay, and diabetes, like crossing the barrier and then going in the bloodstream. Okay, so this is it for like the assignments. If you have any doubt, we can discuss, or we'll discuss the week 5 contents in next class next week. In today's class, if you have any doubt, we can discuss now.
Thank you. te gusta, sí.